So the original title of my talk, that's when I first started as an assistant professor, was really short and snazzy. And as my career has sort of progressed over the last few years, the title's gotten a little bit longer, and by the time I'm full professor, it'll probably take up the entire slide. So I'm here to talk to you about our KATP channels and their role in the nervous system with regards to pain and opioid signaling, and how this may depend on not only potassium channel subtype, but also cellular location, where these channels are actually located. I don't have to do the whole bit about what a KATP channel is and their structure, because I know Colin and William have already given an excellent introduction, so I've cut that material out. You're welcome. But I do want to reiterate, though, that KATP channel subtypes, there are many of these, and the KERS and the SIRS, as I call them, either the KIR or the SIR1's regulatory subunits, their expression depends on tissue type. So cardiomyocytes have a specific type, smooth muscle have a specific type, the pancreas has a specific type, and if you look at any of the recent review articles in KATP channels, it will say nervous system, KIR 6.2, SIR1. That's the end of the story. I guess I'm here to tell you that that's a gross oversimplification, which is not technically correct. It just annoys me just a little bit, maybe more than a little bit. And I just want to let you know that KR subtypes in the nervous system are a little bit more heterogeneous than what the review articles would let you believe. So in neurons, yes, it's true that KR 6.2 and SIR1 are expressed, but KR 6.2 appears to be more important in the peripheral nervous system, whereas KR 6.1 appears to be more important in the central nervous system, including the spinal cord. And SIR1, the regulatory subunits, are more prevalent and appear to be more important in the peripheral system over the central nervous system, and the opposite is true for SIR2. But there's other subtypes of cellular subtypes that exist within the nervous system, and glial cells are definitely there. Shout out to my glia people. Astrocytes are a very important cell type, particularly in pain and opioid signaling. In fact, there's really good data that suggests that KR 6.2 is not in astrocytes. This is going to be largely probably KR 6.1 and SIR2, or excuse me, SIR1 and maybe SIR2. It's kind of up in the air. So essentially what I would just want to reiterate, this is going to come back later in the talk. But the reason why I like KATP channels, one is that they're kind of the Rodney Dangerfield of pain. They don't get any respect. Younger people may have to Google that reference. I might have got somebody to laugh, but they are really almost like a metabolic sensor, and they link metabolism to neuronal excitability. They're also activity is also changed during hypoxia and stress responses, during inflammation. And factors that alter KATP channel function are many, many different things. And then the last reason why I'm interested in KATP channels is because they are downstream of opioid receptor signaling. So they're similar to GERK channels and some other inwardly rectifying channels, is that they are opened upon exposure to opioids due to downstream signaling, which I don't have time to talk about today, unfortunately. And so these are actually T-spiral nerve recordings using an ex vivo skin nerve system prep that we have in our lab. So we largely excise the hind paw and the connecting sciatic nerve, and then we can record single units. And this is an example of a nice happy little C fiber here. And these were animals that had undergone a spinal nerve ligation, which is a model of neuropathic pain that we use in the laboratory. And you can see that upon increasing mechanical stimulation onto the receptive field, the number of action potentials increases. However, after you incubate the skin nerve prep with diazoxide for about a minute or so, the number of action potentials decreases. And we found that this was true in not only C fibers, which are the presumed nociceptors that exist. These responses were also decreased in A delta fibers and also A beta fibers as well, which is actually particularly interesting because these are myelinated fibers and not traditionally associated with pain signaling. Another interesting point that I want to make is that glyburide, or glibenclamide, depending on which side of the pond you're on, they're the same drug, by the way. That took me way too long to figure that out. And antagonists don't really have much of an effect. And so particularly in baseline and nerve injury conditions, the antagonists really don't have an effect, but many of the agonists do. And as another kind of interesting little sidebar, for behavioral studies, we found that the SIR1, more SIR1 selective, and notice I didn't say specific, SIR1 selective agonists were much better at attenuating hypersensitivity than the SIR2 selective drugs. So just kind of keep that in mind for later. And so we mentioned that we're interested in opioid signaling, and several people had found through pharmacological methods that if you give an animal a KATP channel opener and then gave them morphine or some other kind of opioid, these actions would largely be synergistic. And then the opposite would happen if you gave them an antagonist for KATP channels. And so we kind of demonstrated this using a SIR1 knockout animal. These are just global knockout animals that were provided to us by Joseph Bryan. And we found that the wild type animals have a very nice, robust anti-nociceptive response to morphine. And this effect is largely attenuated in the knockout animals. Those are shown in red. And those are pretty consistent across male and female animals and across several different doses. I'm showing you two of them here, 5 and 15 milligrams per kilogram. This is with mechanical threshold testing. In the thermal threshold testing, we didn't see this nice bifurcated response. And so again, that also kind of suggests to us that KATP channels may be particularly functional in these myelinated fibers, may be important for touch sensation. And you probably have already guessed this because you've seen enough pain talks already, but typically when the numbers go up, that means that there's either less pain or anti-nociception, and if the numbers go down for these behavioral plots, that means that there's increased hypersensitivity or more pain. Although I kind of like Dr. Khanna's version of it, the happy face and the frowny face, so I may steal that moving forward. We decided to also use some more genetic strategies in order to kind of look at this more specifically. And so we have a conditional knockout animal. These are SIR1 flox animals. And we looked at specific injection into the lumbar spinal cord. These are intrathecal injections that we did using a neuronal promoter for these particular experiments. Indicated here. And the reason why we tried to do these conditional knockout studies is we got great data from the global knockout animals, but we just wanted to make sure there wasn't any compensatory effects or anything weird like that. And interesting, we did see a sex difference in these animals, which we didn't see in the global knockout animals. And so male animals have greater morphine-induced anti-nociception than in females. So the control animals, the delta Cree, are in the black, and the conditional knockout animals with the active virus are in green. But we did see similar results when we looked at a model of opioid tolerance and withdrawal. And so in order to induce opioid tolerance or morphine tolerance in a mouse model, typically the way it goes is that you give an animal morphine twice a day, morning and evening, for at least 5 days straight. And then you can measure the baseline responses of the animals before morphine every day, and then 30 minutes after. And so you can see here the pre-morphine plots are on the left here, and the post-morphine plots are on the right. And largely, you can see that the delta or the active virus animals, which are the Cree animals, those values are all lower in comparison to the control animals. And so we conclude by looking at the pre and the post-morphine plots was that opioid-induced hyperalgesia intolerance were potentiated in the delta Cree animals as compared to the controls. And this was fairly consistent in both male and female animals. And then we also waited 24 hours after the last morphine injection just to see if hypersensitivity would continue, even in the absence of morphine. And the same was the story was very similar between male and female animals. And we have also done other viral vector studies, and we did find similar data using a short hairpin strategy as well, targeting ABCC8, which is the gene for SIR1. So that was nice and consistent across the board. The next question that we got from the reviewers was, well, when you knock down KATP channels within the spinal cord and the peripheral nervous system, what actually happens to nerve fiber conductance? And so we did some C-fiber compound action potential recordings from these animals. If you do, and I'm not showing you all the beautiful fluorescence that my students spent hours collecting, but the virus will travel down the sciatic nerve and get actually into the nerves that innervate the hindpaws. And so using these C-fiber compound action potential recordings, we largely didn't see a lot of changes in either the threshold in order to induce compound action potentials or the conduction velocity, but we did see a small increase, and this is pretty small, increase in amplitude in the active and the Cree animals. So animals that have loss of SIR1 are largely, their C-fiber conductances are largely kept intact. This is a shout out to one of our sponsors here, Sophion Bioscience. We've been using the Brilliant Flex assay, I believe it's called, looking at potassium channel flux in mouse DRG neurons. And one of the questions that we've been asking for a long time is, well, we know that knocking down SIR1 in neurons has all of these interesting effects related to pain. Which population of neurons are actually driving this response? And it's really hard to do this using patch clamp because I'm not getting any younger and I don't have 50 years to answer this question. So we decided to use an imaging approach instead, just using a spinning disc confocal. And so we also co-labeled using two different markers. One is an IB4 marker, which is isolectin B4, which is typically will label non-peptidergic C fibers, and a cholera B toxin label, which will also label typically larger myelinated fibers. And this is typically what the distribution is pretty consistent between males and females, and this matches the literature quite nicely. And what we found largely were kind of three different bins of responses when we applied, for example, 50 micromolar diazoxide, which is a KATP channel opener. About 40-50% of the neurons would respond pretty robustly to diazoxide, and with at least a 20% response over baseline. We kind of got this intermediate group, which we're not totally sure what to do with, so we just called them the medium responders. And then about a quarter of the neurons don't really respond at all. We have them listed as small or less than 5%, but I think conservatively I'm just going to call them non-responders. And so when we actually looked at the labeling to see where the responses, most of the potassium responses were coming from, we found interestingly that there in the cells that did not label either IB4 or cholera B toxin, which we're presuming are maybe peptidergic C fibers, there's at least a 30% response. 30% of these neurons have a large response to potassium upon application of diazoxide. The IB4 positive group, also there's quite a few neurons that have a large response in this category as well. But the most surprising data, which probably shouldn't have been surprising if I had been paying attention to any of my previous electrophysiology or behavioral experiments, was the cholera B toxin positive subset of neurons. Over half, and sometimes 2/3 of these neurons had a large potassium response. So basically that indicates to us is that KATP channel signaling may be important for not only C fibers, but also myelinated fibers as well. We've been kind of testing this using inflammatory pain models, and largely we're looking at CFA injected animals, and largely I don't want to show you a bunch of pie charts because that's probably super boring, but here we can, just to kind of summarize, the IB4 negative and the cholera B toxin negative population of neurons, so which are presumably peptidergic C fibers, we didn't see any change in the number of the large responding potassium responses. For the IB4 positive neurons, there's about a 20% decrease in the large responses to potassium. And in the cholera B toxin positive population, we can't really find any responsiveness to diazoxide anymore using an inflammatory pain model. So this is obviously ongoing. We've been trying this with a neuropathic pain model. We're trying this chronologically over time to see if it matters when, 1 day, 3 days, 7 days, 14 days out. Is there some kind of temporal resolution that occurs? Much of our behavioral data would indicate that that's not the case, but we're just testing it at a cellular level. So that's to be continued. And then, the last half of this talk is about the difference between the neuronal versus the astrocytic composition of KATP channels. And so somebody had asked me at one point in time if we thought that KATP channels and astrocytes had any role in pain signaling? And to which I responded, I don't really know, because we're not glia people, we don't really know, but it's worth testing. Or we figured, well, we could at least test this pretty easily because we already have the SIR1 flox animals. And so if we just test this with astrocytes instead of neurons, we could answer this pretty easily. And it turns out I figured out why this person was asking me this question, because there was already some data indicating that astrocytes, or KATP channels expressed on astrocytes, maybe did have a role in pain signaling. So what would happen if we removed SIR1 and astrocytes instead of neurons? So for this, instead of using the neuronal promoter, we use a GFAP promoter, so to target astrocytes. And largely, this slide is pretty boring because in naive animals, not much happened. We threw them through a battery of tests using a rotarod, which is a test for animal mobility. We used a burrowing test. We did open field testing. We also did morphine acute dose response curves on these animals. And we also did the morphine tolerance paradigm that I explained to you earlier. So looking at morphine over the course of 5 days. We tested them before morphine application and after morphine application. That's why this graph kind of looks like a sawtooth now, because you're testing before and after morphine, so it goes ear. And the red and the black lines are basically overlapping. So that's not very exciting. And we thought that the story was done until we started to use some injury models. So the first model that we use is a spinal nerve ligation model. I mentioned this model briefly earlier. Largely, it involves ligating or cutting the L4 sciatic, or the L4 spinal nerve. Model of neuropathy may be similar to like sciatica, for example, for the non-pain people in the room. We did the same strategy where we injected viral vectors, either encoding the neuronal promoter or the GFAP promoter to lose expression of SIR1. And interestingly, we tested this at a few different time points, and so this is the data from 2 weeks after spinal nerve ligation. In the neuronal promoter, we didn't really see much of a decrease, and we weren't expecting it because we think that the activity and function of these channels are probably already compromised during nerve injury. But interestingly, on the ipsilateral hind paw, using the GFAP promoter, these animals had increased mechanical thresholds, so indicating that there was less hypersensitivity in these animals. So that was a bit surprising. And it was even more surprising when we did the morphine tolerance paradigm in these animals. So you can see here that the GFAP promoter animals did better than either the control animals, and definitely much better than the neuronal promoter. The neuronal promoter in blue, that data we could have obviously would have guessed because this matches our previous data that I already showed you in naive animals. So it seems like the combination of nerve injury and chronic opioid exposure, the difference between these two become really, really apparent. And then the next thing that we did was we did a dose response curve again on these animals before we did the morphine tolerance paradigm, and then again after. And the idea here is that the dose response curve should shift to the right? So the same doses of morphine that worked really good before morphine tolerance shouldn't work as well after morphine tolerance. And you can see here that there's a shift to the right for the black animals, which are the control vector animals. So these are the delta Cre animals that don't express an active Cre. It does also shift to the right, definitely shifts to the right for the neuronal promoter animals, which we could have totally expected based on our previous published data. However, the red animals, the GFAP promoter, there is no shift to the right. So indicating that these animals do not develop tolerance after losing SIR1 and astrocytes. We also decided, since we thought we were going crazy, we would try this using an inflammatory pain model. So again, this is the CFA model of inflammatory pain. CFA, for those of you that don't know, is basically a killed mycobacterium that causes a local inflammatory response to one of the hindpaws. We call this the puffy foot model in our lab because that's what happens to the mice. Largely acutely, when we looked at mechanical thresholds after CFA, you can see that they decrease after injection. It's exactly what you should see. However, there's really no difference in the neuronal promoter or the GFAP promoter animals. And when we did the morphine tolerance paradigm, very, very few differences between the control animals, the neuronal promoter, or the GFAP promoter. So if you're wondering that you're thinking, well, there's not much going on, I would agree, not much going on. The tolerance experiments and the dose response that we did after tolerance was kind of similar. So the GFAP animals this curve does shoot to the right after morphine tolerance, but nowhere near as extreme as with the previous neuropathic pain model. So I agree that there is some tolerance going on here. It's not as extreme as the other groups of animals, but it was still there. So now we have a new hypothesis. So in a naive state, we still believe that KAR 6.2 and SIR1 potassium channels, they are antinociceptive and they contribute to opioid analgesia. And we still think that this is true also during injury models. However, we have some data that I haven't showed with you today that maybe the loss of expression or function could contribute to particularly neuropathic pain. However, what is to be said now about these SIR1 expressing astrocytes? And so maybe remember that in our naive animals, there wasn't much going on. No real changes in behavior, no changes in opioid tolerance or withdrawal. However, when we added on a chronic pain model, and in addition to a morphine tolerance state, we saw our data was much different. So now we have to figure out what's going on here. There is a theory that maybe SIR1 during neuropathic pain conditions can couple to TRPM4, for example, in order to form cation channels. So basically reversing the activity of SIR1. So instead of being a potassium channel, it becomes a cation channel. That data has largely been proposed by Mark Simard at the University of Maryland, Baltimore. However, Colin Nichols has some contrary data showing that SIR1 does not complex with TRPM4 in vitro, and this is not possible. So the last question I'm sure you have for me is, well, is KAR 6.1 involved in this scenario? That is a great question that I hope to find out in the future. And if you have any ideas on how to figure this out, please let me know. There's a bunch of people that I need to thank, but I will plug not just William Coetzee, but also Suman Song at the ION Lab. And if you haven't heard about the ION Lab and you would like to know more, please let me or William know. The amazing people that are in my lab that actually did this work. And I can't do anything without money. And so I will be done here, and I thank you for your time.
Thank you, Amanda. You're the first speaker to come in under 25 minutes. Congratulations. Well done.
Very, very nice talking, very nice data too, Amanda. So from a therapeutic perspective, what would be the ideal, and from a KATP channel perspective, what would be the ideal drug to treat pain, do you think?
Wow. The ideal drug to treat pain if you wanted to target KATP channels. I mean, and this isn't just data from my lab. Other labs have shown that, KAR 6.2 and SIR1 is important for neuron function. And if you open these channels, then they are antinociceptive. So that could be a potential target. Now for the other KATP channel subtypes, that I think is a much murkier question. That is a really good question.
I have a quick question. So if I really understood it, it's inhibit channels in neurons bad, inhibit channels in neurons good for pain sensing.
It depends on what your definition of bad or good is. So if you inhibit KATP channels in neurons, that's bad. So that is pro-nociceptive, and that increases morphine tolerance and increases withdrawal. Inhibit SIR1, at least anyway, and astrocytes, that's bad. That is good. If you inhibit, now you got me all backwards. Inhibiting SIR1 and astrocytes improved antinociception and improves opioid tolerance and withdrawal.
By the way, we have dominant negative subunits that you can express, and that'll test whether it's TRPM4 or not, and prove that it's KATP.
Excellent.
Okay. In the interest of sticking to time, thank you very much, Amanda. That was great.